Hi everyone. Thank you for being here to listen to this talk. My name is Reiman and I'm a Research Assistant Professor at the Toyota Technological Institute at Chicago. And today I'm excited to talk to you about some work involving a deep learning approach for modeling longitudinal microbiome data. For those that may not be familiar with the microbiome, it consists of a community of microscopic organisms such as bacteria, fungi, and viruses. And this community is believed to functionally interact with its environment. When we think about the human microbiome, we know it can be affected by multiple host characteristics, behaviors, and external stimuli that can perturb this community. We can also think about different microbiome communities in human health, each with its own function. In particular, the gut microbiome, which is the combination of the stomach and the intestines, has become increasingly clinically relevant in many aspects of health. Within the last decade, our understanding of how important the gut microbiome's role in human health has greatly expanded. Early studies were able to associate this community to metabolic diseases such as inflammatory bowel disease and obesity, as well as autoimmune conditions such as diabetes. However, recent studies have also illuminated other important roles of the gut microbiome, such as the development and modulation of the central nervous system, the shaping of the immune system in early life, and even having an effect on a patient's response to cancer immunotherapy treatment. However, one challenge in modeling microbiome systems is the large inter-individual variability that we observe. This means that the microbiome from one individual can be very different from another's, even if they are providing a similar function. But because the gut microbiome can be altered and changed, it has become a targetable factor of interest for therapeutic interventions. To facilitate the development of microbiome-targeted therapies, it is necessary to be able to model how the microbiome changes over time given both host intrinsic characteristics and perturbations from external stimuli, such as therapeutic interventions. For this task, previous methods have focused on either network-based modeling, using Boolean or dynamic Bayesian networks, or generalized low-Clavitera models to create a set of differential equations. However, these models are not scalable and suffer at larger feature and sample spaces. This is becoming a problem as technology is now allowing us to sequence at higher resolutions of microbial diversity, as well as to be able to generate more samples at reduced costs. Therefore, we need more scalable models, and for this we propose a framework which we call DiRLaM that combines two neural network models. The first is an autoencoder model that will compress the microbial community into a latent space, capturing intrinsic patterns of the data. It is here within the latent space that we will both interpolate to smooth the longitudinal data, as well as learn how to predict future time points to model the dynamic changes using a second supervised neural network. So let's look at the first network, our autoencoder, which performs dimension reduction in an unsupervised fashion. This means that if we have two microbiome abundance vectors, x1 and x2, we can compress them using an encoder network into the latent representations z1 and z2, and then decode them back out into microbiome abundance vectors x1 hat and x2 hat. At the same time, we will create an interpolated latent point between z1 and z2 as a weighted linear combination of the two with some mixing coefficient alpha between 0 and 1. And we will call this z alpha, and we can decode it into an interpolated microbiome community x alpha hat. We then train this neural network to minimize the reconstruction loss between the microbiome inputs and reconstructed outputs using the Kullback-Leibler divergence since we are using relative abundance values. Additionally, we add a novel regularization on the beta diversity, which is how we describe the distance or similarity between microbiome community structures. Here we are using a specific metric called the Bray-Curtis dissimilarity, shown at the bottom in a reduced form for relative abundance. Simply, you can think of this as two microbiome communities having a distance of 1 if they are completely different, meaning they share no microbial features, and a distance of 0 if they are exactly the same. Using this, we add a regularization to help enforce that the distance between the two microbiome inputs is equal to the sum of the distances between the reconstructed interpolated point and the two microbiome inputs, meaning we want to interpolate in the shortest distance between the 2 points with respect to how we think about distance in the microbiome space. In the second network, we now integrate external factors and host characteristics, shown in blue. And given the latent representation of one time point, we predict the latent representation of the next time point. This can then be decoded back into the microbiome community space, where we can compare how close the prediction is to our interpolated values. It's important to note here that the encoder and decoder networks are not changed in this part of the training, so the only learning being done is on the network in the middle. To evaluate the interpolation and predictive abilities of DiRLaM, we construct a simulated dataset using the R package Microbiome DA-Sim with 100 subjects, each with 100 microbial features and 120 evenly spaced time points. We separate 50 subjects into one group and 50 into another, where there are 10 differentially abundant microbes between the two groups. Additionally, we perturb the microbiome community with 3 different discrete events, each which modify the dynamics of 10 microbial features, some of which are shared across the events. We will use these as our ground truth for evaluations. However, longitudinal microbiome data never looks like this in real life. To better simulate realistic microbiome data, we add increasing levels of corruption by using only a small portion of time points and only a subset of features at each time point, masking the rest. The first thing we evaluated was the effect of the strength of the beta diversity regularization on the interpolations. To do this, we used tenfold cross-validation and trained DiRLaM using the corrupted data. We then take the interpolations of the held-out set and compare the interpolations across the entire time period with the original uncorrupted data using the Kullback-Leibler divergence. We compared using no regularization, as well as values of 0.2, 0.5, 2, and 5. What we observed is that a stronger regularization improved interpolations in higher corruption settings by penalizing large changes in microbiome shifts, but it could also be detrimental in lower corruption settings. Since we cannot know how much noise truly is in the data, we select a value of 2 for future benchmark comparisons. The next thing we evaluate is DiRLaM's interpolation ability. We compare DiRLaM to standard B-spline interpolations, a similar linear interpolation within a reduced space defined by principal component analysis, and a standard autoencoder, which is essentially DiRLaM with no beta diversity regularization. Models were trained in a similar fashion using tenfold cross-validation, using the corrupted data to train the model, and comparing the held-out interpolations with the uncorrupted data. We observed that all methods performed comparable in the lower corruption settings. However, DiRLaM was more robust compared to B-spline and PCA interpolations as the level of noise increased in the data, highlighting its ability to capture the true signal of the data and create smooth interpolations. Lastly, we evaluated DiRLaM's ability to predict subsequent time points. We first compared DiRLaM to a standard approach of using a dynamic Bayesian network, or DBN, on B-spline smooth data. Then, based on the observation of DiRLaM's improved interpolative ability, we train a DBN model using DiRLaM's interpolated values. We then considered that DBNs suffer at large feature spaces, and evaluated how DBNs could perform using DiRLaM's interpolations within that reduced latent space. What we noticed is that with each change, the DBN models improved, showing that dimension reduction can facilitate training of DBN models as well. However, despite the improvements in the DBN models, DiRLaM was still able to outperform DBNs in each instance. Next, I want to focus on what DiRLaM can learn. Without going into too much detail, we will look at 2 different levels of interpretation. The first comes from the supervised neural network, where we can take our trained models from tenfold cross-validation, and for each of the external factors we are trying to model, we can evaluate how important they are by shuffling one factor and comparing the predicted evaluations with the shuffled data to those without it. And if the ratio is greater than 1, then there was a loss in performance when that factor was corrupted with the shuffle. The second comes from the decoder network of DiRLaM, in which we can characterize how each microbial feature behaves with respect to each latent dimension. This can be done simply by decoding the identity matrix and subtracting from each row the output of a decoded zero vector. These can then be clustered to reveal groups of microbes that behave similarly with respect to microbiome community dynamics. Furthermore, the weights of the supervised network can be used to infer attribute scores of each input feature and output feature, allowing us to infer interactions and construct a module-based interaction network. Using these ways to interpret DiRLaM's modeling, we're going to turn to a real-world biological dataset for the rest of the talk. We will be using a longitudinal microbiome dataset from the study by La Rosa and others that looked at 58 infant gut microbiome communities over about 26 time points per subject. It gave a total of 922 samples and 29 microbial features. Host characteristics included gestational age, post-conceptional age, mode of birth, and biological sex. External factors included the amount of breast milk consumed, amount of antibiotics used, and room ID. Within this dataset, DiRLaM identified 4 significant factors: antibiotics, amount of breast milk consumed, mode of birth, and biological sex. We can then take these factors and create our module-based interaction network. DiRLaM was able to learn that increasing breast milk consumption positively affected the dynamics of gamma proteobacteria, which has been observed in previous studies as well. Additionally, infants who underwent vaginal delivery had an increase in actinobacteria, bacilli, and clostridia, each of which has been shown in other studies to be associated with vaginal delivery. So in conclusion, in this talk we presented DiRLaM, a combination of an autoencoder and a supervised neural network for modeling microbiome dynamics. DiRLaM applies a novel beta diversity regularization to help create smoother interpolations, and we showed that DiRLaM improves upon current interpolation and dynamic modeling techniques. Additionally, DiRLaM was able to learn significant and informative factors in real-world longitudinal microbiome data. I would like to thank Dr. Yang Dai and her lab, as well as additional collaborators. And I'd further like to thank you for watching, and if you have any questions, please feel free to reach out, as I would love to discuss my work. Thank you again.